Greetings everyone, welcome to a new episode of MrBioinformatics. My dear beautiful viewer, let me take your hand and let's go on a journey to the year 1869 to Germany, specifically to Tübingen University. There was a Swiss scientist named Friedrich Miescher. At this time, he was studying white blood cells. Someday, Friedrich decided to go to a hospital and ask nurses for something quite unusual. Use the bandaged from infected wounds. While it may seem unappealing, pus is rich in white blood cells, making it an ideal sample for his studies. Friedrich used a special way to open them. He started to treat the cells with sodium sulfate, which helped him to isolate the nuclei. Then he added an alkaline solution followed by acid, causing a chemical reaction that resulted in a solid precipitate. This substance was unlike anything else scientists had seen before. Friedrich called this precipitate nuclein because it was found in the cell's nucleus. The question here, my dear viewer, why was this discovery significant? Unlike proteins, nuclein contained phosphorus, a chemical element not commonly associated with biological functions at that time. Since its role was unknown, nuclein was largely overlooked. At this time, scientists were focusing on proteins, believing proteins were the key to hereditary. As a result, Miescher's discovery went largely ignored for decades. Scientists continued to believe that proteins were truly carriers of genetic information. But why did they think that? Proteins seemed complex, with 20 amino acids, while DNA was just 4 bases: adenine, thymine, cytosine, and guanine. Imagine researchers split into two groups. One focused on proteins as the key to the inheritance, the other on the DNA. For years, this debate raged on as they struggled to understand how genetic information was passed down. Proteins are the most important. They are 20 amino acids, can do many functions, but DNA has strong 4 bases. 4? What's gonna do? Start a rock band? This debate, my dear viewer, went on until 1944, when 3 scientists, Oswald Avery, Colin MacLeod, and McCarty, conducted their experiment at Rockefeller Institute for Medical Research in New York City. It was later renamed Rockefeller University in 1965. These 3 scientists worked with Streptococcus pneumoniae, a bacterium known to cause pneumonia. When mom tells you to dry yourself well after a shower, she's trying to keep you from catching pneumonia. Anyway, during their studies, they injected harmless bacteria with a mixture containing dead, viral and disease-causing bacteria. The harmless bacteria were transformed and become deadly. This indicated that some factors from the dead bacteria was being transferred to the live ones. Here, Avery and his colleagues were asking an important question. Is the bacteria can it change based on something from the dead bacteria? What exactly is this mysterious factor doing that changing? So they decided to systemically remove proteins, RNA, and other molecules from the bacteria, testing each component to see if transformation still occurred. After eliminating the proteins and RNA, they discovered that only one molecule remained capable of transferring the genetic traits. What was this substance, my dear viewer? The experiment confirmed that DNA is the key to genetic inheritance. Let me tell you, my dear, that although the 3 scientists concluded that the molecule was DNA, the scientific community was still little doubtful. So what convinced them? After 8 years, specifically in 1952, American scientists Martha Chase and Alfred Hershey conducted an experiment using a special virus called bacteriophage, a type of virus that infected and replicates within the bacteria. It consists of a protein coat and genetic material, which can either be DNA or RNA. These scientists labeled the DNA and protein of the virus with radioactive markers to see which part entered the bacteria during infection. They discovered that only the DNA entered the bacteria and was used to make new viruses. This experiment provided clear evidence that DNA, not proteins, is the material responsible for transmitting genetic information. In the same year, 1952, specifically on May, British scientists Rosalind Franklin and her PhD students Raymond Gosling at King's College London, they took many X-ray diffraction images of DNA. Finally, after many photos, they captured photo 51, the image that revealed the DNA's helical structure. Photo 51? We all need a friend like Rosalind and Raymond, ones who don't give up after just a couple of shots. This image showed distinct X-shaped diffraction pattern, confirming that DNA was a double helix, two strands twisted around each other. This crucial information greatly aided James Watson and Francis Crick in building their model. And after 1 year, on 25th of April 1953, they published a paper in Nature that revealed the structure of DNA and laid the foundation for modern genetics. And sadly, despite Franklin's crucial contributions, she didn't receive the recognition she deserved during her lifetime. Rosalind Franklin passed away in 1958 from ovarian cancer at the age of 37. There was an opinion that her extensive exposure to X-ray radiation in her research contributed to her illness, although there is no clear evidence directly linking her cancer to her work. In 1962, Watson, Crick, and Wilkins were awarded Nobel Prize in Physiology or Medicine for the discovery of DNA's structure. Now, my dear viewer, we've learned how DNA is structured. But here is a big question. How do we actually read this genetic code? How do we uncover the sequence hidden inside? In 1977, British biochemist Frederick Sanger took on this challenge and developed a groundbreaking method for sequencing DNA. To test his technique, he chose Phi X174 virus. Why? Because it's a small virus with simple genome of just 5,386 bases, which made it the perfect starting point. To begin the process, Sanger and his team started by preparing a single strand of DNA to read. As we know, DNA is double helix with two strands each composed of 4 nucleotide bases: adenine, thymine, cytosine, and guanine. Sequencing typically focuses on one strand because both strands are complementary, meaning that adenine pairs with thymine and cytosine pairs with guanine. The sequence of one strand determines the sequence of the other strand. For example, if one strand has the sequence AGCT, the complementary strand will be TCGA. Then they added a primer. What is primer? Primer is a piece of DNA to act as a starting point for the copying process. Without the primer, DNA polymerase enzyme, which is used to copy the DNA, would not know where to begin. Let's say that DNA polymerase is like a man at a printer who needs a piece of paper to print or copy the DNA. Someone might ask, but why do we need to copy the DNA instead of using more? The reason that DNA strands are too long and complicated to study all at once. By copying DNA, we create smaller pieces that are easier to study. Copying DNA is also important for cell division, making sure each new cell has the same DNA, and for gene expression, where specific genes are copied to make proteins. After copying the DNA, Sanger introduced dideoxynucleotides. What are dideoxynucleotides? These are modified bases that, when added to the growing DNA strand, stop the copying at that specific point. But why did we add it? This caused the DNA strands to end at random points, creating fragments of different lengths. But why do we create fragments with different lengths? These fragments, my dear viewer, represent different parts of the sequence, much like how you can break a sentence into smaller pieces. For example, if the full sentence is a cat eats 7 shrimps, you might get smaller fragments like a 1 liter, cat 3 liters, eats 4 liters, 7 5 liters, and shrimps 7 liters. These fragments represent different parts of the sentence, just as the DNA fragments represent different parts of the genetic sequence. To figure out the exact sequence, scientists used electrophoresis. What's gel electrophoresis? In this process, an electric current was passed through a gel, causing DNA fragments to move. Smaller fragments move faster, while larger ones move slower. Think of it like you're sorting those sentences by their size. A is smaller, so it moves faster, while shrimp is longer, so it moves slower. By looking at the positions of these bands, Sanger's team could figure out the exact sequence of bases in the DNA. This method allowed them to fully decode the genetic code of the virus. In 1980, Sanger won his second Nobel Prize in Chemistry for this groundbreaking work. Let me tell you, idea that Sanger's path to discovering the secret of DNA was not entirely new for him, as he had already used similar ideas in the past. He won the Nobel Prize in Chemistry in 1958 for his work on protein sequences. If you didn't watch our episode about proteins, make sure to watch it after this episode. His famous achievement in this area was sequencing insulin, the first protein to have its full structure determined. Sanger's method involved breaking insulin protein into smaller fragments and identifying amino acids in each fragment. I feel like anytime Sanger's want to solve any sequence, he split it into smaller ones. He used a technique called Edman degradation, which allowed him to systemically remove and identify one amino acid at a time from the end of the protein chain. By putting the fragments back together, he was able to reconstruct the complete amino acid sequence of insulin. Despite its accuracy, Sanger's sequencing was slow and costly. While the 5X174 virus has 5,386 bases, the human genome, for example, has around 3 billion. Sequencing a single human genome took years and cost millions, leading scientists to integrate computational methods. As in 1990, scientists from the National Human Genome Research Institute in the United States, along with collaborators from all around the world, they launched the Human Genome Project. The genome was completely sequenced in 2003. Although the project was originally expected to take 15 years, it was completed 2 years ahead of schedule due to technological advancements and the collaboration of thousands of researchers. Despite its success, sequencing remained costly, as the total cost of the project was about $3 billion, and also time-consuming. This limitation led to the development of next-generation sequencing, or NGS. Unlike traditional Sanger sequencing, which reads DNA one strand at a time, NGS could sequence millions of fragments simultaneously, drastically reducing time and cost. What once took years and billions of dollars could now be done in a single day for a price less than flagship smartphone or $1,000. NGS revolutionized medicine, biotechnology, and bioinformatics by enabling real-time disease tracking, personalized treatments, and ancient DNA discovery. You can explore this concept further in our bioinformatics tutorial on Python and R. Check out the links in the video description. If you'd like an episode about NGS, please write NGS in the comments below. And here we've reached the end of our episode. Thanks for watching. See you next time.